Good morning everyone. Welcome back to my channel Pari's tutorial. I'm back with one more interesting video. Please let's get into the class. Hello everyone. Welcome back to my class. In this class I want to discuss about one of the important biomolecules called carbohydrates. In the last class, I have already discussed about the nucleic acids and their separation and all. And in the last class itself, I have taken a class regarding the prokaryotic cell and eukaryotic cell. So why I have taken this carbohydrates in this class, because in the coming classes I want to discuss about the metabolic pathways that would happen in the cell. So what really happens, what is cellular respiration, and what are the things that are happening in the cell. For that to have a clear idea, first we should have knowledge about the glucose, monosaccharides, disaccharides, polysaccharides. What are those things? Okay, so carbohydrates are one of the four major classes of biomolecules along with proteins, nucleic acids and lipids. So these carbohydrates are either aldehydes or ketones. So aldehydes are with the functional group CHO and the ketones are with the functional group CO. Okay, they are considered to be the most important organic matter on the earth. Okay, so you know that the carbohydrates that is present in the food is main source of energy. So you all know that ATP is the energy currency, right? For ultimately to perform a work we need to have some energy, and energy is generally produced from the cell in the form of ATP. So in this class I want to discuss about the structure and how the carbohydrates are being broken down into monosaccharides for the easy digestion, right? How the intestine gets how it absorbs all the nutrients from your monosaccharides. Why only carbohydrates are important for the source of energy. Okay, so these carbohydrates are built from monosaccharides. I told you they are the small molecules typically containing 3 to 9 carbon atoms. Okay, so whatever we consume, so whatever we take the food, first of all it has to be broken down into simpler molecules for easy digestion. It could be polysaccharides, it could be whatever the food that you take. First you manually broken down into simple molecules like glucose, then it gets absorbed into the blood, right? So that is why glucose is very important for the conversion. So for you to have an idea about the structure of glucose, what are the positions of carbon atoms and hydroxyl groups in glucose, I want to talk about carbohydrates. Okay, so why carbohydrates? So first of all, why carbohydrates are preferred? Carbohydrate serves as energy stores, fuels and metabolic intermediates. The second reason being, ribose and deoxyribose sugars form the structural frame of RNA and DNA. I have already told you the DNA and RNA are made of ribose and deoxyribose sugar. In the last class, in the previous classes, I have already discussed the structure. You can go through the class if you want to go for the structure of DNA and RNA, where I have also clearly shown the picture of the ribose sugar and deoxyribose sugar. Okay, and the third, polysaccharides, you all know that cell walls, in the last class itself I have discussed that cell walls are mainly made up of polysaccharides, and so polysaccharide is also a carbohydrate. Okay, what is cellulose? So cellulose is also present in most of the cell walls, right? So it is considered to be the most abundant organic compound in the whole biosphere. Okay, and fourth reason being, carbohydrates are linked to many proteins. Okay, it also helps in cell to cell communications and interactions. So that is why carbohydrates is considered to be an important source of energy for all of us. Okay, so what are monosaccharides? So monosaccharides, I told you, these are the simplest form of carbohydrates. Whatever the food that you have consumed, it has to be broken down into simpler substances for easy digestion. So monosaccharides are the simplest form of carbohydrate. Whatever that you take, it has to be broken down. Okay, so I told you they are either aldehydes or ketones that have two hydroxyl groups. Okay, so here I want to show you what are monosaccharides and what are the types of monosaccharides. There are mainly two types. They are called as aldoses and ketoses. Whatever the substances that contain aldehyde as their functional group are called aldoses, and which contain ketone as their functional group are called ketoses. Okay, I will tell you what are those. So the aldehyde, the main parent compound for the aldehyde is, or you can call it the simplest form of aldehyde, aldose, is glyceraldehyde, and the simplest form of ketose is dihydroxyacetone. Okay, so I will show you from why L and D are made. So you often being confused with what are the forms of glucose, why it is called L form, why it is called D form. So for you to talk about it, I want to talk about the glyceraldehyde. Okay, so glyceraldehyde has, see, I will show you what happens here. See, this here I have given you the reason. So glyceraldehyde has got a structure where it has an aldehyde group here, and it has got only one asymmetric carbon, right? So only one asymmetric carbon in glyceraldehyde. So based on the position of hydrogen and hydroxyl groups on both sides, they are mainly differentiated into D-glyceraldehyde and L-glyceraldehyde. So D-glyceraldehyde and L-glyceraldehyde are different only in the C2 position, like second carbon position of your aldehyde. So these are considered to be the stereoisomers, okay, and these both are the mirror images of each other. D-glyceraldehyde and L-glyceraldehyde are the mirror images of each other, and they are also called enantiomers. Okay, so this is your aldehyde group. See, in this D-glyceraldehyde you have got H in right position, and here you have hydroxyl in this position. It's exactly the mirror image of D and L. That is why these are called the stereoisomers or enantiomers. Okay, they are the mirror images. So that's why I want to show you. Okay, I told you more than two, like 3, 4 could be pentoses, hexoses, like all like simple monosaccharides. It could be 4, 5, 6, 7 carbons are called tetroses, pentoses, hexoses, and heptoses. I told you. Okay, so here I have given you a structure where I want to discuss with you. See, this is the glyceraldehyde form. Okay, I given you. This is a 3 carbon compound, right? It's a 3 carbon compound. Now it has got the same asymmetric carbon is shifted away from your aldehyde group. Sorry, this is shifted away from the aldehyde group. See, you can notice it clearly. This is your aldehyde group, and this is your second carbon, this is your third carbon, and this is your fourth carbon. This is called erythrose. 4 carbon compound of aldose is either erythrose or threose based on the position of the hydroxyl groups, but this won't change. Okay, this particular asymmetric carbon shifts their position away from the aldehyde group. Okay, and the carbons keep on adding here. See, you can notice here in ribose, which is a 5 carbon compound, it starts with aldehyde. This is your second carbon, this is your third carbon, and this is your fourth carbon. So 2 carbons have already attached, and this is called ribose. This keeps moving away from your aldehyde group. In the same way, here arabinose you can observe where there are 2 carbon atoms in between the aldehyde group and your asymmetric carbon. So based on the position of your carbon atoms, these structures keep on changing. Okay, so you got idea that these are the 4 carbon compounds, and ribose, arabinose, xylose, and lactose, these are all the 5 carbon compound. Okay, now let's talk about the 6 carbon compound, because out of all these aldoses, the ribose, glucose, mannose, and galactose are more present, like abundantly found. Out of all these, you can see ribose in most of your RNA structures, right? And you can see glucose in animal cells and all. Even galactose and mannose are also abundantly found. Okay, most available aldoses. So see, ribose is a 5 carbon, and here you got 2 different types of aldoses, which are D-allose and D-altrose. Okay, and arabinose has given rise to glucose and mannose. Okay, here comes the main important discussion. See, this is a D-glucose and this is a D-mannose. Here in the second carbon, the difference is there. See, this is your aldehyde group, the first carbon. The second carbon, the arrangement are 2. See, COH and OH, these are 2. I can call them as epimers at C2 position. These glucose and mannose are epimers at C2 position of your structure. See, the rest of the being the same, only one carbon has got a change, which is called an epimer at C2 position. Okay, the same way, let's now compare glucose and galactose. See, in glucose and galactose, you can have one difference where you can come across the C4 carbon. the carbon in the fourth position. See, 1, 2, 3 and 4. At the fourth position, the arrangement of hydroxyl and the OH groups are in the opposite direction, which says glucose and galactose are the C4 epimers, like fourth carbon epimers. Glucose and mannose are carbon 2 epimers at their second position of the carbon. So they might be asking you what are epimers, at what position the glucose and galactose are different, at what position glucose and mannose are different. So it's better you should have a clear idea. And by the way, these are all the open form of structures. And what important point I want to talk to you is glucose in its ring form is more stable than the glucose in the open form. I will let you know why in the next slide itself. Okay, so glucose are always whatever the monosaccharides, all these mannose, glucose, galactose, everything should be in a ring form. Then only it would be easy for the reaction to occur. If it is an open form, it could leads to skiffs, base and all. I will let you know in the next slide itself. So hope you got an idea what are aldoses. So next go for the ketoses. So I told you the parent compound, the simple monosaccharide of ketoses is dihydroxyacetone. They got a group called CO. So you know that acetone has a CO group in it, right? So and based on it you have got erythrulose, ribulose and xylose. So erythrulose is again a 4 carbon compound, and D-ribulose is again a 5 carbon compound, and xylose is a 5 carbon compound. Okay, so now, and again it has differentiated into sucrose, fructose, sorbose and tagatose. Okay, out of which the fructose is considered to be the most abundant ketoses. Out of all the ketose, fructose is the most abundant ketose. Okay, so hope you got an idea. Like these are all the aldoses and ketoses. The pentose and hexose cyclize to form furanose and pyranose. Yes, so pentose is like I told you, pentose like ribose example. So pentoses like ribose and glucose are like hexoses like glucose, they form a ring, okay, within themselves. Why? Because when you when the glucose is in the linear form, or when it is cut open, this linear form is not a stabilized one. It won't function properly, and it's not preferred. Okay, so the open chain forms should need to be cyclized. Okay, for the stabilization they have to form a ring like structure, forming a bond within the molecule itself. So they need to form furanose ring and pyranose ring. Okay, so for example, take an example of glucose. The C1 carbon contains the aldehyde group, CHO group, and that particular C1 carbon of glucose would form a chain like structure and reacts with the C5 group of hydroxyl group of the same glucose within it. So it leads to the formation of a small ring like structure called pyranose ring. Okay, in the same way, we are taking an example of a ketohexose. Like ketohexose example is a fructose, right? So if you take fructose as an example, the second carbon compound of the keto group will react or would form a bond with the hydroxyl group present in the same compound within the molecule itself. Thus leads to the formation of a furanose ring. Okay, so that would be the formation of pyranose and furanose, because the stabilized part is more important for you to reaction the occur. Okay, what are common disaccharides? So the disaccharides generally consist of two sugars that are mainly joined by a glycosidic linkages. So as you already know, there are three abundant disaccharides present. They are sucrose, lactose and maltose. So sucrose is a disaccharide where it is hydrolyzed into a glucose and a fructose. Okay, in the same way, the lactose, commonly called as milk sugar, is also a disaccharide. When it is hydrolyzed, it would be giving into one molecule of glucose and one molecule of galactose. In the same way, maltose also, it is a disaccharide. When hydrolyzed, it gives you two molecules of glucose units. Okay, now this is the common structure that I want to talk to you. Right, just now I have drawn this because it would be easy for you to understand which bond is made. Okay, see, sucrose I told you, this is the structure of glucose. Okay, this is the structure I will tell you. Right, okay, this is your structure of your glucose molecule, and this is the structure of your fructose molecule. this is the alpha position of sucrose is bound to or made a bond with the glycosidic linkage with the beta position of your fructose. So the name comes as, I told you, pyranose ring is formed by glucose and furanose ring is formed by fructose. So the name is derived as alpha D glucopyranose, one first position to second position carbon atom with the beta position D fructofuranose. That is how you have to write it. Okay, I told you glucose pyranose ring and fructofuranose ring. Okay, so glucose forms a pyranose ring and fructose forms a furanose ring, forming a ring like structure. Okay, so this is alpha D glucopyranose with 1 arrow to 2. This is your fructofuranose. The same way lactose, I told you lactose is also disaccharide where it is hydrolyzed into one molecule of glucose and one molecule of galactose. Okay, so see, lactose is linked with the first carbon of your galactose with the fourth carbon of your glucose. So that is why the bond is like, it is beta position. This is the beta position of your galactose. So this is got a glucosidic bond with the alpha position, forming a 1,4 alpha. Okay, and the same way maltose is again hydrolyzed into two molecules of glucose, and see, it is the easiest structure being D-glucopyranosyl with one carbon and alpha position linked with the fourth position of your glucopyranose. So both are glucose having glucopyranose rings. Okay, these are the disaccharide structures. So they may ask you what are the bond linkages between the sucrose. They may ask you linkages of lactose and maltose. This is why I have given you the detailed structure of the glucose, maltose and lactose. Okay, sucrose is cleaved into components, monosaccharides, by an enzyme called sucrase. So sucrase acts on sucrose. For that to happen, you should have a presence of sucrase, maltase and lactase. You should be knowing about lactose intolerance, right? What is that? In adults, many people in adults and in some kids also, you will come across a term called lactose intolerance, because the milk sugar, the milk is made up of lactose. Okay, that lactose, for it to get digested, for the milk to get digested, they have to have an enzyme called lactase. If they don't have the enzyme lactase, they can't break the lactose molecules into simple monosaccharides like glucose and galactose. So the people, whoever is not having the lactase enzyme in their body, they are called lactose intolerance. Okay, this is sucrose, and sucrose is acted by sucrase enzyme which would produce glucose and fructose. Lactose is acted by lactase enzyme which would break down into glucose molecule and a galactose molecule. And maltose is one important disaccharide where it is broken down by maltase into two molecules of glucose. So all these enzymes must be present in human body for the easy digestion of your disaccharides. So sucrase, maltase and lactase are mainly present on the outer surface of your epithelial cells lining the small intestine. Okay, and now what are polysaccharides? So these are a long chain of your oligosaccharides. They keep like, for example, if you take a cellulose contains a long chain of glucose units repeatedly. Okay, the cellulose is the best example for that. Okay, large polymeric oligosaccharides found by linkage of multiple monosaccharides are called polysaccharides. They play a role in energy storage and maintaining the structural integrity. I told you cell walls are all made up of polysaccharides. It gives you a rigid quality and a strong structure integrity, right? If all the monosaccharides are same, yes, they are called homopolymers. Okay, the most common homopolymer in animal is glycogen. Okay, now you all know that whenever we are at rest, the glucose is stored in the form of glycogen. Okay, whatever that we are taking in when we are at rest, the whole thing gets converted into glycogen for the further usage, right? Whenever we require ATP, like whenever we need energy, whatever is stored in the form of glycogen would be converted into glucose again. This is called as gluconeogenesis. Okay, this is called as gluconeogenesis. Okay, glycogen, if all the monosaccharides are same, Like I told you, polysaccharides are nothing but multiple units of monosaccharides. If all the monosaccharides in a polysaccharide chain are the same, they are called the homopolymers, and the best example for the homopolymers is glycogen. Like all the units in glycogen are made up of glucose. Okay, the all monosaccharides that are present in glycogen are glucose units. So it would be easy for us for the breakdown of glycogen into glucose whenever we need energy. Okay, so glycogen is a very large branch polymer of glucose residues. Most of the glucose units in glycogen are linked by alpha 1,4 glycosidic bonds. As I told you, the glucose, even in the maltose, if you take an example of maltose where two glucose units are linked by alpha 1,4 glycosidic linkage, in the same way the whole polysaccharides of homopolymers are all linked by alpha 1,4 glucosidic linkages. Okay, next, the nutritional reservoir of carbohydrates in plant is starch. I told you, in animal cells glucose is stored in the form of glycogen, whereas in the plant cells the glucose is stored in the form of starch, and the starches are all two forms. They are amylose and amylopectin based on the branching type. Already I told you it's an alpha 1,4 linkage between the glucose units. If it is like alpha 1,6 linkage, that is a branch type of linkage. 1 and 6th carbon of your glucose molecule. It is called a 1,6 linkage. If those are present, they are called amylopectin. It is stored in the form of amylopectin. If amylose is only stored, it only contains alpha 1,4 glycosidic linkages of all glucose units. If it is amylopectin, it contains glucose 1,4 linkages and also few alpha 1,6 linkages also. Okay, that is, they are called the branched type of starch. Branched type of starch present in carbohydrates and unbranched type. Okay, so more than half of the carbohydrates ingested by the human being is starch. Both amylose and amylopectin is hydrolyzed by alpha amylase secreted by salivary glands and pancreas. Right. When we take the food, you know that salivary glands has got amylase in it. So this amylase would help you for the first primary digestion happens in your mouth because of the enzyme called amylase, where the glucose residues are broken down into simple residues, right? The polysaccharide of amylose is converted into glucose molecules. So these are the linkages till now. Okay. You're wondering why only glucose is chosen for your monosaccharide, and why is it is on the only simple sugar for everything to be converted. So whatever you eat, it is immediately converted into glucose and it is immediately absorbed into the blood, right? So why only glucose? So the reason being, glucose is one of the several monosaccharides formed from formaldehyde under prebiotic conditions. This is the first monosaccharide that is formed from formaldehyde under prebiotic conditions. Okay, and one more thing, glucose has a low tendency relative to the other monosaccharides to non-enzymatically glycoside proteins. This is important. Okay, because glucose is stable in its ring form, right? It is easy for the glucose to react in the ring form. When it is in the open form, when the glucose is in an open form, and it immediately reacts with the amino group of your protein. Proteins are made up of amino group, right? Amino, you have amino end and a carboxyl end, right? The amino end of your protein reacts with the open chain form of glucose, and that leads to the formation of amino keto linkage, and that is called a Skiff's base. And this Skiff's base do not function properly. This is not specifically bound. They are non-specific modified protein. The protein is completely modified because of your open chain form of glucose. So glucose, if it is in the open chain, it is not in an active form, and it leads to Skiff's base formation, which is not functioning properly. So when the glucose is in the ring form, it would be having high energy molecule, it is having high stability, and it will help obviously will reduce the tendency of modifying the proteins. So that is what is important, right? The protein should never get modified when it is added with the carbohydrate, or when it is added with the glucose. So modification of that particular protein is completely low when it is glucose. That is why glucose is taken as the best monosaccharide for the absorption into blood. So hope you got an idea about the carbohydrates. As I keep discussing about the pathways, and please get to know the details. Please, I'll be linking all the structures in the description box. I'll just go through that links, and also I want to tell you which book I prefer for biochemistry, or especially the carbohydrates and all the other metabolic pathways are Stryer Biochemistry. That is, the Stryer is the best book for you to follow. If you want to go through, you can go through in the ebooks also. You can have those links and you can read from them also. Okay, so as of now, this is the topics that I have covered. In the next class I will see you with the metabolic pathways. So thank you and have a nice day.